Good morning, everybody. If we can start, please take up your seats. My name is Ian Craig. I'm the president of IFAC, and we've just got a very short ceremony before we start with the plenary this morning. I've got the pleasure of making another IFAC Advisor Award this morning. And as I said in the opening ceremony, the success of our federation is partly due to the fact that the leadership of IFAC can rely on the advice of advisors who have exceptional experience and wisdom in IFAC matters. And it's now my privilege to appoint such an IFAC advisor whose wise counsel will greatly benefit the future leadership of IFAC. Can I ask Professor Lina Gutzala to come to the stage, please? Before I make the award, I just want to tell you a little bit about Professor Gutzala's background. Lina Gutzala is a faculty member in the Institute of Dynamic Systems and Control at ETH Zurich. And since 2012, he's the rector of this institution, and he's also at the moment the president-elect of ETH. After receiving his mechanical engineering diploma and doctoral degree from ETH, he held several positions in industry and also in academia, including being R&D team leader at Zulzer Brothers and Honda visiting professor at the Ohio State University in the US. He received the IEEE Control Systems Magazine Outstanding Paper Award, the IEEE Industry Award for Excellence in Translational Control Research, the SAE's Arch T. Colwell Merit Award, and the Rolf R. Teeter Educational Award, the IMECI's Thomas Hawksley Medal, and the Crompton Lancaster Medal, and the Energy Globe Award. He is a fellow of IFAC and of the IEEE, and a member of the Swiss Academy of Engineering Sciences. The citation for this Advisor Award reads, "For outstanding contributions in managing the finances of IFAC." Lina Gutzala. Thank you very much. I'll now hand over to our session chair for this morning, Yanan Zaitun.
Good morning, ladies and gentlemen. My name is Janan Zaitoun from the University of Reims, and it's my great pleasure and honor to chair this plenary session. Just before introducing our speaker, I'd like to make two announcements. The first announcement is that someone has lost the watch in the meeting of Technical Committee on Energy and Power Systems, so I've got it here. And the second announcement from the Congress organizers saying that airport transfers can be booked at the registration area. So, our plenary distinguished speaker is Professor Mustafa Khammash, and his talk is related to, don't have the title there, Cybergenetics, feedback control of living cells at the gene level. Professor Khammash is Professor of Control Theory and System Biology at the Department of Biosystem Science and Engineering at the Swiss Federal Institute of Technology in Zurich. He received his PhD in control theory at Rice University, Houston in 1990. Then from 1990 till 2001, he was on the faculty of electrical engineering at Iowa State University in Iowa. In 2001, he joined the mechanical engineering department at the University of California in Santa Barbara, where he served as the director of the Center for Control, Dynamical Systems and Computations. And from 2006 till he joined ETH in 2011. So Professor Khammash is a fellow of the IEEE, IFAC, and the Japan Society for the Promotion of Science. He is the recipient of the National Science Foundation Young Investigator Award, the Iowa State University Foundation Early Achievements and Research and Scholarship Award, the ISU College of Engineering Young Faculty Research Award, and the Ralph Budd Best Engineering PhD Thesis Award. Professor Khammash.
Okay, thank you for this kind introduction. Congratulations to Lino. You know, he's my future boss at ETH. Thank you all for being here this early in the morning. And thanks for Ian for inviting me and giving me the opportunity to tell you about my research and that of my group. I actually joined ETH about 3 years ago. And when I joined ETH, I was thinking about starting a new wet lab, which is something I hadn't done before. And so I wasn't sure whether I should do it or not. I finally decided, you know, why not? We'll give it a try. I had been working in the systems biology area for a while, and I think it was time to move closer to the applications. And I think it was one of the best decisions I've made. So what I'll tell you about today is basically our research over the last 3 years at ETH, and the title, which I will explain, my slide is Cybergenetics, the feedback control of living cells. Okay, so let me get started. So our goal, in a nutshell, is to manipulate the dynamic behavior of living cells using real-time dynamic feedback control. So why would we want to do something like this? Well, there are various reasons. I mean, there's applications in biology, trying to develop and design tools that probe biological dynamics. There are applications in biotechnology and therapeutics, and I'll outline some of those. But really, I wouldn't be honest if I tell you these are the reasons that drove us. We just were interested, at the time we were working on the chemical master equation, and we were fed up trying to do analysis for everything. We thought maybe it's time to start doing some control. This is how it started, and this all came after the fact. So in his 1948 book, Norbert Wiener presented a vision where control and communication for the animal and machine are unified under the same framework. And he called this area cybernetics, which means the art of steering. But at the time when Wiener did this work, the structure of the DNA hasn't been resolved yet, and the applications were limited mostly to neuroscience and some other applications. But today, more than 60 years later, we have a very powerful tool for manipulating the genome, and it is possible to control processes at the cellular level that weren't even dreamt of at the time of Wiener. And so in order to keep the connection with Wiener's vision and to separate this from some of the other research that's going on in the area of synthetic biology, I thought it would be good to give this emerging area a name, and I thought cybergenetics would be a good word to keep the connection. So I will refer to this work that I will present to you as sort of cybergenetics. So what has motivated the development of this area? There are several enabling technologies that made this possible. Nowadays, especially over the last 10 years or so, there's been more advanced methods for stochastic analysis of biocomputational or networks. There are, as I mentioned, modern, powerful genetic techniques for manipulating genomes, and I'll try to explain some of those, and we will use them for the work that I will show you. But there are also new ways for stimulating cells. So this allows us to apply control signals, and I particularly mention optogenetics and microfluidics. And finally, it is also possible nowadays to measure variables from the cell and measure variables from populations of cells as well as single cells. And the technologies I'm referring to here is flow cytometry and time-lapse microscopy. Okay, so here's a quick outline. The first couple of minutes I will give you a brief view of basic cell biology, at least extracting the aspects of it that we will use for the presentation. And then I will talk about two different modes for control. One I will call in silico control, where we use computers to do the control, and the other one I will also discuss in vivo control. And finally, I will present a theoretical framework for biomolecular control design that I think would be useful for future work. Okay, so let's begin at the very beginning, the single cell. So if you look inside a single cell, it's a very crowded place, and it's filled with proteins, various enzymes, various nucleic acids, mRNA, DNA, and so on, lipids. And these constituents, what I call cellular species, they move around due to their thermal energy, and they interact with each other, forming complex networks that are required to carry out biological function. Okay? There are various aspects of these reactions or these interactions in the cell that are important. One of them is the randomness that's inevitably involved due to the thermal motion of these molecules. But the other one is that these molecules are present in a wide spectrum of copy numbers. So some elements can be as low as 1 copy, so for example 1 or 2 genes per cell, maybe a handful of messenger RNAs per cell. For proteins, it could be in the thousands or tens of thousands. So these aspects offer various challenges when we try to model and control these systems. Now, one hallmark of cell populations is its variability. So even if you take a collection of cells that have exactly the same DNA, and you grow them in exactly the same environment, you will clearly see cell-to-cell variability. And the origin of this cell-to-cell variability is due to some of these factors that I mentioned, low copy numbers, random fluctuations in the cellular constituents. But it is something that we have to contend with, both in the analysis and in the design. So if you look at the abundance of a given protein in a cell, it might look something like this. So it's a piecewise constant because molecules come in whole numbers. And then if you look at a neighboring cell, which has exactly the same DNA, growing in the same environment, at the same temperature, seeing the same nutrients, you might see something like this, or a third cell, something like that. And so when we try to model these cellular dynamics with stochastic models, what stochastic models will give us is something like this. So you can see this, for example, dark line that represents the concentration of a protein in a given cell. But actually, in many this may be adequate for some applications, but in many other applications where these fluctuations are important, a probabilistic approach is required. And I will mention this in my talk a little bit further on. Okay, so I will tell you, maybe give you a brief review of one of the most fundamental chemical processes in nature, that of gene expression. You may or may not remember it in high school, but you do it a billion times a day. So this is how gene expression works. So gene expression is the process by which proteins which are encoded in the DNA are transcribed and translated into functional molecules, functional proteins. And so everything starts at DNA level, where, oops, let me just go back. Everything starts at the DNA level, where the protein is encoded. And then this here is a site called the promoter site, where a molecule, a big molecule, initiates the transcription process, which you can think of it as a big reading head crawling on the DNA, transcribing the sequence of nucleotides into a transcript, which we call the messenger RNA. Okay? So this is the process of transcription. Messenger RNA could degrade, or it could be translated by other machines called ribosomes into functional proteins. In this case, it's proteins called RSC. So this goes on in every living cell, gene expression. And the gene expression is actually highly regulated for some genes, not so much in others. And the regulation actually depends on binding of other proteins into this site upstream from the gene. Okay, this is fine and well, but how can we even learn about this? Well, one clever idea would be to fuse to the sequence of that protein a sequence for another protein called GFP. And then when that protein is expressed, it actually forms this, forms the so-called fluorescent protein. So this fluorescent molecule, the fluorescent molecule here, when you shine a laser with a certain wavelength, it emits light of a green color that you can actually detect. Okay? So this simple idea revolutionized molecular and cell biology. And actually, because it allows biologists now to look at cellular structures and watch essentially biological function in real time. It also made a huge impact for those of us who want to do modeling, because it allows you to quantize the effect or the amount of proteins that are present in a given cell. And so think about this: if the more abundant that protein is, the higher the intensity of the fluorescence. And if you can pick up the fluorescence, then you have an idea of the concentration of that protein in the cell. So widespread is the use of this green fluorescent protein that the people who discovered it and later developed it were awarded the Nobel Prize in Chemistry in 2008. That's Shimomura from Japan and Martin Shelfie and Roger Tsien. And one of the contributions of Roger Tsien is to further mutate this green fluorescent protein so that it can respond to different light wavelengths. So now you can see red, cyan, and other colors, allowing us to actually encode multiple different proteins in the same cell. So we can actually detect the concentration of these proteins in the same cell at the same time. So now going back to the picture of variability, if you look at let's say a flask full of these cells, because of the variability that I told you about, cells will have different fluorescence intensities. And now, if you take a sample from the cell culture and pass it through a flow cytometer, which is basically a microfluidics device that allows you to shine a laser at these cells one at a time, and then picks up, you have light detectors that pick up the emitted fluorescence. And then you can look at about several 100,000 cells per minute using this flow cytometer. And by plotting the distribution of light intensities, we have a snapshot of the probability density function of the distribution of that protein in the cell population. And this is what you see over here. So it's a very invaluable tool, and it is a tool that we will use for measurements throughout this presentation. Okay, moving on. So the first thing I would like to tell you about is this computer control of gene expression in bacteria. It's joint work with Andreas Milis Argaitis and Mark Rulan in my group. But before I do that, I would like to review some earlier work that I've done with a former student of mine, Hannah El-Samad, who's now a professor at UCSF, and my colleague at ETH, John Ligeros. And this is how we got started on this idea of cellular control with light. This work was published about 3 years ago. But the idea was, what if we can use light signals to actuate gene expression, and then by measuring gene expression and putting in a feedback loop, we can regulate the amount of protein that is being expressed to any desired set point. So this was the main idea here. And we have shown earlier that we can do it theoretically, and then we moved this light system into yeast cells in Hannah's lab at UCSF, and we were successful in carrying out the first control process of gene expression, so first computer-controlled gene expression process. Now I'll tell you a little bit more about the details of this computer control system, but I will use a sort of a more recent system that we have done, this one in bacteria. So here's the idea. So in this particular case, we have a light-responsive bacterial cells. But if you shine green light, about 535 nanometers, that has the effect of self-phosphorylating this membrane protein And this green dot that you see here is a phosphor group that gets passed on to another protein in the cell, a CCAR, so response regulator. And when this protein gets phosphorylated, it dimerizes and it basically turns on gene expression. Okay? In other words, by shining green light, you activate a certain protein that allows the initiation of gene expression. And the protein that's encoded in this particular case, which is interesting doing the feedback, is a green fluorescent protein, like the one I showed you in the previous slide. Now, when you shine red light on these cells, this has the opposite effect. So it is not completely understood, actually, the biology of this process. This was taken from another bacterium, moved into E. coli. But it is believed that shining red light makes this protein CCAS act as phosphatase, taking away the phosphor group and in the process deactivating gene expression. Regardless of what's the explanation behind it, it really works. So, you shine green light, you have gene expression, and with red light, you don't have gene expression. And the nice thing about this particular system that's not true for the previous year system, that it allows you to apply signals at very large ranges. So you can do graded control signals. The other one was so sensitive that it acts like a switch, either on or off. So we ended up using pulses. So to develop this further, one of the problems we had to overcome is the large variability that's involved in any work, actually, in cell biology. And we've discovered early on that things are not very repeatable, and so we have to do something about it. So as engineers, we thought we can try to control this environment as much as possible in order to get repeatable experiments. And so one of the first things we did is we built this device called the turbidostat, which allows you to control the cell density. So the cells are growing when you give them food, and if you don't dilute the culture, the density of the cells becomes so high they move into another state, stationary phase. So we would like to keep them growing at a particular rate, and to do that we had to devise a closed-loop feedback system whereby we measure the cell density by shining light that has nothing to do with the activation in a different wavelength, and then closing the feedback loop by applying a different amount of fresh culture that keeps the cell concentrations constant. And only then can we apply our light system to it. The other thing is that these experiments take a long time. They take hours. In the case of yeast, maybe 8 hours per experiment. And in the previous work, this was done manually. So somebody every so often, every 15 minutes or half an hour, has to come in, do the dilution, take the sample, and so on. And that introduces a lot of errors and introduces a human in the loop. So we wanted to build a system that's completely automated for sampling. And using a connection of valves and pumps and computers, we were able to do that. The idea is that you take a sample as frequently as you want in the tube, you follow it by air, and then every time after you send the sample to the flow cytometer, and then turn on the flow cytometer with a clicking program that allows it to take the measurement, and then you wash the tubes again. And so this was done automatically every time you do the sample. So now we can do actually much more frequent samples. We can also start this process late at night and come back in the morning having all the results. So this was actually a big improvement for the well-being of the graduate students. Once you have the measurements, one can actually use these flow cytometry measurements back into the computer. We implemented various algorithms, and I will tell you about some of them, to track the protein concentrations to a certain set point. So the idea is that the biologists would be sitting behind their desk, and if they would like to increase the protein level, they would dial in a certain amount of protein. This would be the set point, and then watch the computer generate these light signals to adjust the protein levels to a fixed amount. Okay? All right, so this is one of the experiments that was done a few weeks ago, where we were able to use a simple PI control feedback loop to do this control. And so the idea is that we have a set point here, and then we're starting the cells from this level, and we bring the cells up to this steady state. And one thing you can notice is that this is actually not a simulation. This is biological data. These are biological measurements. So I think by taking out the human factor from the sampling and the errors in dilution and so on, one can get very accurate measurements from these cells. So we can reach the steady state, and then at this time we've applied a very large disturbance, and you can see that the controller recovers from the disturbance. We actually wanted to run this experiment for 1 more hour at the end to show that you have a full steady state, but then Windows decided it was time to upgrade, so the whole thing crashed. And this thing takes a while to set up, so I can't show you, but I think you can see that this thing is heading in the right direction. The other way is that feedback is really required here. So one can think about applying perhaps open loop, and there have been some papers recently in very well-established journals proposing that one can use this light in open loop mode. And the idea here is that it simply isn't possible. And so just to show you, one of the applications we want to use this for is as a signal generator. And we have here a sinusoid. We can use a model predictive controller to track. And then when you can see the control signal here that gives this tracking. Now when we apply the same exact control signal to the culture the next day in open loop, what you see here is the green trace. Okay? And the same thing for a step response. So you use feedback control to do the step tracking, take the control signal, apply it the next day in open loop mode, and you start getting large errors. So to understand why this happens, actually we just decided to run step response experiments just in the open loop, different days, and this is what you see. And I'm talking about these are cells that have this exact same DNA, the exact same growing conditions, same nutrients, and they're even with the turbidostat in operation. There's very, very large sensitivity to these cells. So I think feedback control is indispensable for regulating gene expression. So when we published the yeast work at first, so let me just give you some applications actually, or potential applications. So one possible applications would be to put these cells in bioreactors to be able to regulate byproducts, perhaps limiting byproducts, or maybe one would like to limit biomass, so you can regulate the growth with light while keeping the product production at a high level. We're currently talking with Novartis to try to put some of these systems in CHO cells, these are hamster egg cells, for antibody production. The other applications we have in mind actually is as a tool for basic molecular biology research. So by using light, we can probe biological networks at a certain node and then look at the response, perhaps doing a frequency response, if you will, for a given chemical reaction network, and in the process trying to get better understanding of how these cells work. So this would be a tool for network identification. Okay, so when we first published this work in 2011, this yeast cells with John Ligros and Hannah El-Samad, we were very pleasantly surprised that we actually got quite a bit of press coverage. This happened to be the first time anybody controlled gene expression with a computer. And the interesting thing, Jason Palmer, the editor, gave this a nice name. He called these cyborg yeast. And so this has captured the imagination of a lot of people who had various different applications for this kind of work. And just to show you some of the things that people have done, so one proposal here is interface with Jean-Luc Picard with the Borg. So this is the cyborg cells. And then some more serious ones. But then this is also one of my favorites. Cyborg yeast could idiot-proof your home brew. So this is the biotech application. Okay, very well. So the next thing I would like to do is to tell you about how we can take this control into the cell. So we want to move the controller from the computer inside the cell. So building a control system inside the living cell is joint work with Gabriele Lillacci in my group and Stephanie Aoki. So the in silico control model I described to you has several advantages. It allows you to move sensing, actuation, and control outside the cell, where you can do a lot more. So you can actually implement very complex controllers. You could do MPC, you could do optimization, you could do Kalman filtering, and you can change the program very easily, just with the press of a button. So this would take several minutes to change a program. Compare that to biology, and one of the things I've learned about the biology, and at least the experimental aspects of biology, is that it progresses at a geological time scale compared to all of the other things we can do. And so to think about changing the program would actually require minimum 1 week, and that's just changing one parameter, and I'll explain this a little bit later. But one of the other advantages with this in silico control is that that you have the control system has access to the entire population, because you're doing these measurements with flow cytometer, so you know the distributions of the cell, so you can actually take these measurements and you have more information to use in feedback. On the other hand, compare that with what I would call in vivo control, where you would build this controller inside the cell using genetic techniques, and then each cell will have its own controller. Now this has other advantages. So each cell is autonomous, so it has its own controller. You don't have to keep track of it with light, and that turns out to be actually a significant advantage over the in silico control. But each has different applications. And so this is what I would like to tell you about next, is how we put controllers inside living cells. And so to do that, I would like to review some basic techniques from molecular biology. So here again is the cell. And in this time, I'm identifying two different pieces. The big thing is the chromosomal DNA. That contains the program for the whole cell, life of the cell. And there's another piece of DNA called the plasmid. Okay? And there are several of them usually in typical bacterial cells. And it also is a DNA. So if you look at a slice from a plasmid, you can see it's actually double-stranded DNA. And it has a sequence between one of 4 nucleotides, AGCT, that encodes different proteins. Okay? So when this program, you should think of it as running a script. So if running the chromosome is like the whole computer program, the plasmid is like a short script. And it actually is not a bad analogy. In fact, this particular plasmid we used in my lab has about 11 kilobits. Okay? So we could put 4 genes on it, so we can encode 4 genes on it. Typical E. coli bacteria has something like 4,000 genes. So this is the scale at which we're operating. And the nice thing about these plasmids is that they allow you to reprogram the cells. So it is possible to synthetically design the sequence that you want, sequence of DNA that you want, into a plasmid, and then put it into a living cell, and then the cell will execute whatever program is there, so that you can execute the genetic code that you have built in. Of course, this has been going on for quite some time, and I'm not going to spend any time telling you about the process, the cloning process that's involved, but the important thing I would like to say is that each time you make any change, it takes 4 days, and then 1 day for testing, so 5 days. Even though it's possible, it is very time-consuming. So that adds new challenges to the design of the circuits. In any case, once you insert this plasmid into the cell, it interacts with the cell machinery, and then the encoded genes can be expressed. Perhaps the resulting proteins, what I show you here in blue, can act to turn on or turn off another gene downstream, and this creates this gene regulatory network. Okay, so this is the main idea. You have to create these genes and put them on a plasmid in a way to carry out the function that you would like. So what kind of things can we vary in this process? What can we control? So this is various different locations on a typical gene. I'm not going to go over any of those, but you can see certainly the reporter and where the fluorescent protein can sit, and the protein of interest will be encoded, but various other sites for control. And let me just show you what manipulating some of these sites does. So you can alter the transcription rate, the mRNA degradation rate, the translation rate, fluorescent protein, protein degradation rate, and the protein condition by manipulating these sequences. Now, unfortunately, it is not quite possible today, at least with today's technology, to know exactly what sequence corresponds to what rate. So if you tell me I would like to double the transcription rate, I don't really know how to go in and change the sequence so that the rate is actually doubled. But there are a few genes that have been very well characterized, and so people use them repeatedly throughout this area of synthetic biology to build circuits. So this is something that obviously requires a lot of new research, and we're not there yet. Again, this has been going on for several years now. People use these ideas to build devices and build modules. This is the area of synthetic biology. Some of the famous examples that people have worked on are toggle switches, oscillators, and OR gates, and so on. And so, and now these actually gadgets are trying to find their way into biotechnology and into therapeutics. And there have been various work in vitro and in vivo circuits that have been built. In the area of control, there hasn't been too much yet. There's been nice recent work from Murray's group and from Eric Klavin's group and various other studies that are theoretical studies to show how these things actually can be used for the purpose of control. Okay, so I give some references here. In the next part, what I'd like to show you is one of the circuits that we have built and just got working, a new tunable integral controller in E. coli. Okay, so here's how it works. We first have the device a plant, and the plant is essentially gene expression, this particular protein called LAC-I. And so LAC-I is expressed here, and LAC-I is fused with a red fluorescent protein. We would like to quantify its amount and make sure that the circuit works, so we add a red fluorescent protein. Now, LAC-I tetramerizes into this tetramer of 4 copies, and this is going to be, so just the whole population of LAC-I is our output. Now, to do the feedback control, LAC-I itself inhibits the expression of another protein called ERA-C, which in turn is fused with a green fluorescent protein. Now, this ERA-C is going to be our control signal. Okay? So whereas LAC-I is the output, ERA-C is the control signal, which feeds back into the and activates the production of LAC-I. So we have a negative feedback circuit here. And then in order to control this circuit, we have introduced this inducer called IPTG that will allow us to determine the set point. So IPTG is actually a modified sugar that doesn't get metabolized by the cell. Think of it as a sweetener that the cells don't use. And so its concentration remains constant throughout the whole experiment, but it's a very nice way to allow us to talk to the cells, to send signals to the cells. And in this case, we want to use it to fix the set point for the output that's of interest. Okay, so why does this work like an integral feedback? Let's see here. If you think about it, if the rate of production of this ERA-C is approximately a function of Y and R, the IPTG, then the concentration, the U, the controller, is the integral of this f of y r, and if this thing is stable, we can actually get a fixed point. Now the thing is, I say approximately because it's never possible in biology to get an exact integrator. The integrator is always going to be leaky for no other reasons, because these are growing cells, and as they grow, that creates dilution, and this dilution factor can actually look like a leakage term. And I'll show you more in terms of the dynamic equations in a little bit. But in any case, so this is the circuit. One of the things about the circuit that it has to be tested, and so in order to test it, we have devised another module and put it also on the plasmid that's called the disturbance module. So this disturbance module allows us to introduce a disturbance to actually verify and test that our circuit is indeed rejecting disturbances. So in this particular case, this is driven by another sugar called rhamnose that turns on expression also of LAC-I. So that gives us another mode of introducing more of LAC-I so that we can watch our circuit do the disturbance rejection. Okay, so we built this into E. coli. And we also had another circuit that acts as the open loop. allows us to produce the LAC-I just in open loop mode, as well as the same disturbance module, so that we can make the comparison between the two. Okay, so just a brief description of how this could work. Now I removed the mRNA from the equations just to simplify, so we have the concentration of the control signal, the protein RC, and the concentration of the output LAC-I. So the production rate here of the control signal is dictated by, is a function of the output LAC-I through this Hill type function. And then we have active degradation introduced, as well as the degradation of the protein, the natural degradation of the protein plus dilution due to cell growth. And in the case of the synthesis rate for the LAC-I, again, this is the function, first order function for the synthesis rate minus the degradation. Now, in order to make this act as much as an integrator as possible to reject disturbances, these terms have to be as small as possible, so delta 1 has to be small, so we can control the protein half-life and we can make it very stable. And in this case, exactly what we do. And so there's only dilution involved. And in this case, if K2 is smaller than U, this here is like a constant, and so we have this U dot essentially a function of Y. And the dynamic ranges of U and Y must be very carefully matched in order to get this to work. I'll show you in phase diagram for this. So here we're looking at the two null lines that result, and their intersection is going to be the operating point. So this will be the amount of output we see, the LAC-I, the red protein, and this here would be the input signal, which would be green. And by adding or removing IPTG, we are able to actually shift this curve to the right or to the left. Okay? So higher concentrations of IPTG move this to the right, lower concentration of IPTG moving this to the left. And you can see exactly what that does to the set point, or to the fixed point. So for high amounts of IPTG, you expect that you are going to have a high amount of output. For low IPTG, you have low output, and the control will follow suit. Now, because of the saturation in the control signal, there is a practical maximum amount of protein that can be tracked. Okay? That's dictated by this saturation curve. I'm going to call this Ymax. Okay, what happens when you introduce a disturbance? Well, when you introduce a disturbance, either through adding another source for this output or maybe taking out that protein, that has the effect of shifting this curve to the right or to the left by amount proportional to that disturbance. Okay? And if this control system is designed well, if it indeed has an integrator, or at least a leaky integrator, we would expect to have a very high gain here, so that introducing disturbance doesn't change the output all that much. So it will have a good disturbance rejection property. Okay? Of course, we would expect that to deteriorate as we start operating more in this region. Okay, so this is what we did. We put this in E. coli, and I'm about to show you the step response of E. coli. You're the first to see it. So here's what the step response for E. coli looks like for different amounts of IPTG. So what we do is we grow these cultures overnight, these cells overnight, and we take the culture, dilute it for 3 hours, let the cells reach their steady state with no IPTG, and then we introduce for different cultures different amount of IPTG. So that's a different set points. And then we watch how we take samples over time to see how the input changes and how the output changes as a function of this. So this is no IPTG, so this is reaching a set point, steady state. And then when you introduce 2 micromolars of IPTG, you get this set point. And then this here is for 5, 10, and then finally 30. Now, the amount of output is not a linear function of IPTG, but it's a very repeatable function. So we don't have to worry. We could just create a table lookup matching the amount of IPTG with the set point, and it's extremely reliable. This thing is repeatable, so we've repeated this many, many times. It's very highly repeatable. And you can see these error bars are actually mostly due to experimental error, measurement error. This was done manually, not with our turbidostat. But so there's manual dilutions involved. And if you look at the control signal that actually achieves this output, you can see it here. So again, very, very repeatable. So the next experiment we decided to do to test this is the following. So starting with different initial conditions, so we have cultures. These are 4 different experiments. So in each experiment, we have culture that has a different initial condition for the output. And then we take each one of those cultures and then introduce them to the same amount of IPTG to see if they would actually converge to the set point corresponding to that amount. And sure enough, in this case, these cells do converge reliably to this output. And you can see as well the control signal. And this can stay for hours. So finally, to look at disturbance rejection. So the most challenging part of disturbance rejection is at the higher parts of the signal. So when the output is at its largest, and you can see that from the phase diagram over here, this is when the gain is not going to be so high, and so you would expect the disturbance to introduce large deviations in output. So just to show you, in the steady state here, we've applied different amount of disturbances for the uppermost curves here, and for 10%, this is a percentage of the output, we get basically almost perfect disturbance rejection. We cannot detect any change in the output in the closed loop compared to the open loop, which actually responds by this amount. So we're using the open loop as a benchmark to measure the amount of disturbance that's being added. So in the case of 40% disturbance, also we cannot detect any change in the output for the closed loop, and we get about 83% disturbance rejection at the highest amount of input, so disturbance of 80% of the output. So this is at the extreme level. And then looking at the 30 micromolar, the highest curve up here, we see a similar behavior. So in this case, we have 80% disturbance rejection at 10% of the output, 83% rejection at 35%, and 71% disturbance rejection at the higher end. And that's what you would expect, actually, for this circuit. And things look quite a bit nicer in the lower ends. So anyway, this is just a proof that this actually can be done and can work. Let me just tell you a little bit, I think, about the practical challenges. In other words, why it took 225 plasmids to do this. What that means is, think about it, 224 failed attempts at making this work. And every time you change one parameter in one of these genes, transcription rate, translation rate, it takes a week. It takes 4 days to do the cloning, 1 day to do the testing. So you have to do things in parallel. You cannot do things sequentially. And but just to outline some of the challenges, well, first of all, dynamic range matching. So you have to make sure that these things intersect in the right range so that you get the right disturbance rejection that you want. So you have to adjust the parameters carefully to do this. But also measurement. Measurement was a big problem. When you add the fluorescent protein, it changes the binding rate of the protein attached to it. So it doesn't always, so this is not a desirable thing, but of course it's a necessary thing because we have to see what's going on. You add a protein, it's going to affect, you add a fluorescent protein, it will affect the function of the protein it's attached to. So we have to take that into account. And then these cells are growing in log phase, and because we have to do dynamic measurements, there isn't a whole lot of fluorescent protein accumulating, so we have to increase the signal so that we can see more green or more red. Okay, so that led us to produce large amounts of U, large amounts of Y. So we wanted to increase. So we had to use large control signals just so that we can see. And also we wanted to stay in the deterministic domain so that we can use these ODEs. So we used the large plasmid copy numbers. So about 50 plasmids per cell here. So that's how many copies of that gene there is in this cell. And then testing poses another problem. We had to essentially devise a different set for testing that took almost as long, maybe in some cases longer than the actual circuit. But you have to have it in order to demonstrate that this is working. And finally, there's this cell response. So the cell is not going to sit there idle, let you play around with its programs without responding. So in some sense, producing these large amounts of proteins does impinge on the metabolic processes of the cell. So you're taking away materials and resources from these very, very highly optimized organisms. They're optimized for growth. And so the cells will be affected if you overproduce these proteins. So you have to strike a balance between, producing large signals, but not too large so that the metabolic burden will be too large. There's also problems of crosstalk, but also mutations. So this is an interesting thing. So when you put these programs into the cells, they usually have random mutations. And if your circuit affects the cell metabolism in a negative way, any random mutation that knocks your circuit out will produce a cell that is more fit. And so its progeny will take over the population, over time. And see, in fact, it's even worse than this because the more stressed these cells are, the higher the rate of mutations, And so we found this problem especially with the open loop strain. I mean, even after one day, these cells mutated our circuit out, and now they're happily living without the cell. You can see it. You could sequence the genome before you do the experiment, and then several hours later, and you can see they've taken out a whole gene, or maybe there's a single point mutation that renders the circuit ineffective. So there's this critical balance between producing the amount of protein that's required to do the control and not to overdo it so that the cells will evolve it out of the system. So far, our circuit, we've run this over a whole day, several times, maybe 20 or 30 times, different times, and it hasn't evolved, but we're very sure that if we let it grow, perhaps for a week or 2 weeks, that eventually the cells will evolve this out of the system. So this is a good point to leave this topic with a quote by Louis Pasteur, who says, Monsieur, ce sont les microbes qui auront le dernier mot. So in this case, it is the microbes that will have the last word. And so these are some of the challenges that we face in the case of synthetic biology. And it's not unique to us, it's everybody who works in this area faces these issues. So this is motivating us to look at the next topic. And the next topic is control with a few molecules. So why did we have to use large copy numbers to impinge on this metabolism of the circuit? Could one get away with another approach? And I would like to argue that actually it is really not necessary to do control with large copy numbers. And the thing is, control is not about energy, it's not about materials. Control is about information processing. And so what you would like to do is you would like to process information, taking the sense signals and then produce an effective output. And that doesn't need to have a lot of resources. So what I will describe to you next is a framework for doing just that. Now, of course, anytime you do control at low copy numbers, you have to deal with the stochasticity questions. Okay, so this is joint work with Corentin Briand and Ankit Gupta from my group. We have a chemical reaction network, so we'd like to talk about this chemical reaction network and the stochastic aspects of it. Some of it would be low copy numbers. So we imagine we have n different species, x1 up to xn, and these are modeled as discrete random variables. So we have a vector of random variables. These species interact with each other through m different reactions. Every time one of these reactions fires, of course, it changes the copy numbers of the species according to the particular stoichiometry of the particular reaction. And since we have m different reactions, we have a stoichiometry matrix with m different columns, S1 up to Sm. And finally, the reaction rates actually forms what's called the propensity vector, W1 up to Wm. Each reaction has its own propensity function, which reflects the rate of firing of that reaction and is dependent on the state of the system. You know, the more molecules you have, the more reactants you have, the higher the rate, the higher the chances that this reaction will fire. In a stochastic setting, these things act as intensity or transition intensities for the Markov chain. So just to give you an example, very, very simple example of how this works, if you look at these two reactions, A plus B gives C with the rate K1 and C degrades with the rate K2. The stoichiometry matrix, the first column of stoichiometry matrix, reflects the change in copy numbers after one firing of the first reaction. So after first reaction fires once, you have one fewer molecule of A, one fewer molecule of B, thus the -1, and then one extra molecule of C, so you have a +1, and similarly for the second reaction. Now, W of X reflects the rate of the reaction. If you think about the mass action kinetics, the rate of the first reaction is proportional to the product of the concentrations, X1 and X2, K times X1, X2, and the second one is just a linear function in concentration of C. And so this is quite straightforward. If you think about this stochastic aspects of it, this represents a continuous time discrete state Markov chain. So it's something that we're quite familiar with. How about tools for analysis? There are various tools. One can write down the forward Kolmogorov equation and get the chemical master equation, but I'm not going to do that here. Instead, I'm going to just show you the moment dynamics. In this case, one can write down the differential equations, in this case, ordinary differential equations that describe the evolution of the moments. And in vector form, this is what they look like. Now, I want to distinguish between two different cases, one in which the propensity function is linear, or affine in this case, of the state, so Wx plus a constant, or quadratic propensity functions. Okay? In the first case, actually, these differential equations can be readily solved. They turn into linear ordinary differential equations. In fact, they're differential Lyapunov equations that can be solved easily. Unfortunately, in the case of quadratic propensity functions, these moment equations are not closed. And this is not hard to see if you think about what happens if you're looking at the second moment here. If this is quadratic, this depends on the dynamics of the third moment, but then you have to write the differential equation for the third moment, which depend on the fourth moments and so on. So you have an infinite set of equations and you cannot close it. There are, of course, approximation methods, but they're just that, approximations. We would like to avoid this moment closure issue altogether in doing control. So anytime you want to do control at the stochastic level, at the low copy scale, you have to deal with stability. Anytime you want to do control at any scale, you have to deal with stability. In this particular case, we have to come up with a special notion for stability that is suitable for these stochastic networks. And that notion is ergodicity. So starting with n reactions interacting with each other through R reactants, just like I showed you before, we will call this interconnection of reaction networks ergodic if there exists a unique stationary distribution to which any initial probability distribution converges to it in the limit. Okay, so there's a unique stationary distribution. So that's what we would call as, so think about this as stability of a fixed point in the deterministic setting. This is the analog here. Okay, very well. So the control problem in this case could be stated as follows. We want to start with an open loop stochastic chemical reaction network, has n different species, just like I showed you before. And since that's going to be the open loop, we would like to control it by introducing new reactions. Okay? So these new reactions that we would introduce could lead the system to become unstable or not ergodic. So in order to frame the problem theoretically, we would like to design a new set of reaction networks, a molecular controller, if you will, such that when you augment it to this open loop set of reactions, the entire set, the closed loop, is ergodic, but also the regulated species, in this case, let's say the nth species, approaches a desired reference in the mean. Okay, so that's the tracking, the steady state tracking question. So we want to do ergodicity plus steady state tracking. Okay, Now, the controller, we have two species to deal with. There's an actuated species, X1. The first one will just renumber things so that the first one is the actuated one through which the controller action will take place, and the last one is the measured species, which is also the regulated species. And the controller I would like to propose has the following structure. It's got two new species, Z1 and Z2, or Z1 and Z2, as we say in Europe. And then these four different reactions. So these reactions, I'm just going to briefly explain the logic. Oops, watch how this thing works. The first reaction acts as a reference to allow us to do tracking. The second reaction as measurements, so the rate of production of Z2 depends on the concentration of this measured species, proportional to the measured species, Xn. And then there's this curious reaction. It's a bimolecular reaction where Z1 and Z2 annihilate each other at a constant rate, eta. And then finally, the feedback term where you can actually produce action on X1 depending on the amount of controller species Z1 here. Now, think about intuitively, the degradation rate of Z1 and Z2 is the same because they basically annihilate each other. So they have the constant degradation rate. In order for this closed-loop system to be stable or ergodic, what has to happen is that their production rate must be the same. Otherwise, one species would just shoot off. Okay? And so this only happens when mu is equal to theta times xn, so the production rates will be the same, degradation rate will be the same, and we get stability. Well, that just means that xn is equal to mu over theta, and that's our set point. Okay? So this is some form of a stochastic integral feedback controller that one can implement with reactions. And I have to say it's related, but not the same as some reactions that were introduced by Eric Clavens for the deterministic control case. Okay, so how does this work? Now I'd like to present you with a general result that allows us to use this. So starting with a general chemical reaction network that's shown here in the cloud. Xn is a measured species, X1 is a control species, and then what's shown on the left here is these reactions that I showed you, these reactions here. So this is how the feedback works. And then I'd like to present this particular result that allows us to do the analysis. So what this result says is, okay, in this case, I'm starting with an open loop which has affine propensities. Okay, just for simplicity, one can introduce into it non-affine propensities as long as they appear in a certain way, but let's just focus on the affine propensities. Now notice that as a closed loop system, the moment dynamics are not closed for this. So we cannot really look at the moments because the feedback controller has bimolecular reactions leading to this moment closure problem. So we have to look at other means. Okay, so what this result says is if you take the stoichiometry matrix times the propensity matrix, S times W, and then form this positive linear system. This positive linear system describes the evolution of the moments just for the open loop. Okay, and since this is closed because it's just the open loop, and the result says that if this open loop system is asymptotically stable and output controllable, okay, then we're guaranteed that when you put these things in closed loop, the reactions will be ergodic, the system will be ergodic, the first and second order moments will be bounded and converge, and finally we have our tracking condition met. So the expected value of xn converges to mu over theta as time goes to infinity. Now this is a very general result, and if you think about what it means, it basically says, the first conditions, first of all, asymptotic stability, output controllability for this system just means that transfer function is not zero, so this is a very mild condition. It basically means if you close the feedback loop, if you think about the linear ordinary differential equation analog, if you take a dynamical system, linear dynamical system, and close the feedback loop with this integral feedback controller, that will always be stable, regardless of the parameters, as long as the open loop is stable. Clearly not true in the case of deterministic dynamics, but definitely true in the stochastic case. So I'm running out of time, but I'm just going to I wanted to show you a brief idea of the proof, but I'm going to skip that. The idea is that to just create a Foster-Lyapunov function and then show that it satisfies some positivity conditions and the derivative or the drift is negative. And the thing is, the idea is quite simple, but the construction is rather tricky. You have to look at partitioning the state space and then defining, in this case, linear Lyapunov functions or piecewise linear Lyapunov functions, and then demonstrate that by properly picking the parameters, you can actually meet these conditions. And the results just come as a direct application of results from the 90s from Sean Mine and Tweedy. Mine and Tweedy's book. Okay, just a quick application just to show you how this works on gene expression. So this is a standard gene expression with our circuit, integral controller. The result basically says that the closed loop system is going to be ergodic regardless of the parameters. So we have of the controller or of the plant, and it achieves asymptotic tracking as well. Okay, so that's quite a nice result. We've done some simulations to show this. So here are some examples where you can see that in the steady state, the output is actually reaches the desired set point very nicely in the mean. Now, we cannot compute these things directly because, as I told you, the mean equations are not closed. But we can do Monte Carlo simulations, and we can look at the averages. And in this particular case, you can see that the output tracks very well. This is what the other guys are doing. And just to demonstrate robust, okay, here's another important point. If you look at the low copy numbers, if you look at the controller species, just for single cells, is extremely noisy. Okay? Even though the mean is very well behaved, this is very noisy. If you look at a single controller, in this case Z2, these molecules are fluctuating between 0 and 1 molecules. Okay, so very, very low copy number control. So you may say, well, this is absurd. Nobody wants to do control with one molecule. But actually, it is not so crazy. About 10 years ago, we studied this heat shock response system in bacteria. And one of the main molecules that is responsible for feedback control is a sigma factor called sigma 32. And when we looked at the abundance of sigma 32, at least free sigma 32, it was doing exactly that. It was going between 0 and 1. Occasionally, you get 2 copies per cell. Okay? So that's how some of these cells do their control, is very low copy. I'm not saying that you actually need to go back to 1 copy to be able to do control. What I'm saying is that one can do very effective control even with molecules as low as one single molecule. Okay, just to demonstrate the robustness, if you change the parameters, let's say at time 25, we double the amount of Kp. Again, looking at the blue trace, which is the output, you have a transient and followed by the same steady state, the same for different outputs. Okay, this is, so tracking works extremely well in this circuit. Okay, this is the final part, just a very interesting thought experiments I would like you to think about. Suppose we take these cells with this controller, and then these are identical cells, they have the identical parameters, the same even initial number of molecules. And then we do this experiment, we break the cell boundary, we break the membrane, and then we make one big cell out of this multitude of cells, okay? So the reaction rates haven't changed, the concentrations haven't changed, everything remains the same, except now we have a bigger cell. And this is the domain of deterministic dynamics. You know, so you have large copy numbers now, large volumes. This is where deterministic equations presumably apply. Okay, and I would like to compare the 2 scenarios, you know, this population of cells versus the deterministic case. In the stochastic case, we've already seen this. This is very nice. This is the mean of the output x2, and it converges to a constant. But when you look at what happens in the deterministic case, this is what you see. The same system, the same initial conditions, the same molecules, represented deterministically, is oscillating, okay? So in the stochastic case, it's very nicely behaved. This is the concentration or the expected value of the concentration. So to get a little bit deeper insight, let's look at the moment equations. So in the first case, these are the deterministic dynamics. We have a second order linear system in feedback with a nonlinear control system which has 2 states, Z1 and Z2. And then we can write down the moment equations. We can't solve them, but we can write them down, okay? And if you look very carefully and compare these two equations with each other, they're only different by one term, and that's this covariance. So this is what the noise is doing, or these fluctuations are doing. So these fluctuations are adding an extra feedback term that's actually helping us stabilize the feedback system. So far from being a nuisance, these fluctuations are actually helping stabilize an otherwise unstable system. And this works regardless of the number of parameters, or regardless of the values of the parameters. So these covariances are always going to have a stabilizing effect. Okay? It's a very curious thing where stochastic fluctuations are actually helping stabilize the system. Okay, and this is my final slide, just some applications of this sort of cell control. Our vision is to actually put these into in mammalian cells, and we've started doing that already for several months now, to put in the integral feedback controller. And the idea is that you would have these cells detecting some, let's say, blood sugar, and then based on the concentration, turn on gene expression according to the control law that I just described to you, and the process secretes something like insulin, for example. This is not so far-fetched. There's a paper from a group in BSSE 2 weeks ago who showed how one can do something very similar to this, and showed how that it actually works in mice. Okay? It's from the group of Martin Fussenegger and DBSSE. Okay, just to summarize then, I've showed you that real-time feedback control of living cells is actually now possible. In silico control is where you do the control with a computer, contrasted with in vivo control, where the controller is actually implemented inside the cell using genetic techniques. And I've also introduced a new integral feedback genetic controller that has been built and tested in E. coli and shows to achieve tracking, adaptation, and tunability. Okay? I mentioned several applications. I've also introduced the beginnings of a theory for doing control with low copy numbers, which I think is one way to help avoid this metabolic burden issue in synthetic biology. There are various opportunities to address control problems in this area. And this is one field where we can actually make a difference, and we can make, at least make steps to the betterment of humankind, which is what IFAC promotes. And I think the skills and the know-how and the tools that our field and control uses can have a lot of benefit, and actually is very appreciated in the synthetic biology and control community. And so I just to tell you, one of the most gratifying comment I got on this work came from a biologist who wrote a famous book on the physical biology of the cell. So I had presented the brief introduction of control and showed how you can use it to do this cyborg yeast. And then he came up to me afterwards and he said, you know, seeing how you can make these cells dance makes me feel more comfortable about flying on an airplane. And that was, I think, the best compliment I've ever got. So that I would like to end, just a final acknowledgement for the people who did the work and current and former collaborators. And I would especially like to acknowledge ETH in Zurich, without whose support none of this work that I talked to you about today would be possible. Thank you very much for giving me an hour of your time. Okay.
Thank you very much, Professor Khammash, for this outstanding and inspiring speech. If you have any questions, you are most welcome to come and discuss it during the break with Professor Khammash. And I'd like you to join me one more time in expressing thanks to him for this outstanding speech.